Good day, everybody. This is Chris with the Ancient Scholar. I hope this video finds you all doing well. Today, what I'd like to do is I'd like to spend a little bit of time talking about mutations that we're seeing in SARS-CoV-2. SARS-CoV-2 is, of course, the virus that causes COVID-19. Now, I've talked about mutations in prior videos, but this video is going to be a deeper dive into where some of these significant mutations are occurring, what the results of some of these mutations may be, and how we can conceptualize mutations and understand their impacts. And of course, how are some of these mutations named? What is the naming scheme and what's going on there? I think this will hopefully illuminate things a lot better for some of us, particularly those of us that may have questions or are very interested about this topic. Before we take a deep dive into viral mutations, I want to spend a little bit of time talking about how proteins are made because this fundamentally involves proteins. Specifically, I'm going to be talking about the spike protein because this is where many epitopes for antibodies exist, and this is an incredibly important target when it comes to vaccine development and just the human immune system in general. So it will be a focused discussion around the spike protein, but again, spike protein is a very heavy hitter when it comes to the virus docking to the cell, attaching to the cell, and fusing with the cell. The spike protein is an incredibly important component of all that, and so from an infection standpoint, this is an incredibly important target. All right, so let's talk a little bit about proteins really quick. So as we know, proteins come from amino acids. So amino acids are basic building blocks for proteins, and these are basic biological molecules, and there are essentially four major categories of basic biological molecules. There are, of course, the carbohydrates, the simple sugars. These are sometimes referred to as monosaccharides. There are the lipids or the fatty acids. There are the nucleic acids, which includes DNA, RNA, and then, of course, there are amino acids, and the amino acids are the fundamental building blocks for proteins. So essentially what happens is amino acids combine to form peptides, and then when you have long chains of peptides, you have what's called a polypeptide. And then what happens is there are side groups on these amino acids that begin to interact, and that causes this polypeptide chain to begin folding into the protein, and this is known as the conformation. And some proteins can have multiple different conformations. So when we look at the basic amino acid structure, is as follows. It is a molecule that at its core has what's called an alpha carbon, and that alpha carbon is very important because there are three major groups that then come off of that alpha carbon or covalently bonded to that alpha carbon. On one end, you have a carboxylic acid group. This is a COOH, a carbon that is double covalently bonded to two oxygens, and then there's a hydrogen that comes off of one of those oxygens, so it's essentially an alcohol side group. And that's known as a carboxylic acid. And the carboxylic acids are incredibly important, incredibly common organic acids. And then on the other side of the molecule, you have an amine or a secondary amine that is actually derived from ammonia. And of course, ammonia is NH3, that is a nitrogen that is covalently bound to three hydrogens. And then when we replace one of those hydrogens with a covalent bond to make a primary amine, we call that an amine group. And that is, of course, where the term amino acid comes from, is the amine group and the carboxylic acid. And then the third major component is this R group here. And this R group, just kind of think of it as everything else. And essentially what you get coming off of that central alpha carbon, in addition to the carboxylic acid and the amine, is you get this R group, and it can be something as simple as a methane, a CH3, but it can also include more complex chemical side groups. So that's the basic structure of amino acid. And to build a protein, essentially amino acids are combined. And what you have is you have the amine component of one amino acid is chemically combined to the carboxylic acid component of another amino acid. So this is amino acid 1 here, and this is amino acid 2 here, and it has R group 1 and R group 2, and that could be whatever. There could be dozens of different types of amino acids with different R groups. And you can see I've got carboxylic acid here and the amine here, number 2. And so there are enzymes that, of course, catalyze this. And essentially what happens is you pull a water molecule out of these two groups here, and that's referred to as a dehydration reaction. And a general rule of thumb is in biochemistry, dehydration reactions result in the combining of things. We call this a dehydration synthesis. And then hydrolytic, when you break water molecules, hydrolytic reactions generally result in things being broken down. And so we call that hydrolytic cleavage. So a dehydration synthesis reaction occurs, and ultimately what happens is the nitrogen in the amine component of one amino acid is then covalently bound to the carbon of the carboxyl group in the other amino acid. And you have one hydrogen atom still on the nitrogen and one oxygen atom over here on the carbon. And of course, the other hydrogen that was attached to that nitrogen and the hydroxyl group that was attached to the carbon got pulled away to form a water molecule. And that's where the water comes out. And you now have what's called a peptide bond that results. And now you have a polypeptide. You have amino acid number 1 has been bound to amino acid 2. And then what can happen is as you get more and more amino acids binding, right? I get a polymer of amino acids, and of course we call that a polypeptide. Those side groups, those R groups, then can begin interacting with each other, and you can get various kinds of intermolecular forces. You can get hydrogen bonds and disulfide bonds and van der Waals forces and induced dipole interactions. And essentially what that does is that causes that linear, that polypeptide, which is thought of as kind of linear, begins to fold and twist in on itself as those side chains begin to interact with one another. And that's where you get the secondary, tertiary, and even quaternary structure of the protein. So the amino acids, the polypeptides rather, these polymers begin folding in on each other to produce the protein. And of importance is you will always have with this long polymer, you're going to have one part of it is always going to have a carboxylic acid at one end, and the amine at the other end. And so this end, even when this becomes a protein and it folds into its conformation, you're still going to have one end of it that's going to have a carboxyl, and this is sometimes referred to as the C-terminus. And then the component of the protein where you have the amine is sometimes referred to as the N-terminus. Okay, so that's the basic understanding that we need, or that's the basic information that we need to understand mutations. All right, so let's talk about mutations in a little more detail. So mutations to viral RNA, because we are talking specifically about SARS-CoV-2, these mutations to the RNA, because remember, the RNA, specifically the messenger RNA, through some complex mechanisms, through RNA-dependent RNA polymerase, which is an enzyme and a protein that the virus has, kind of goes against the central dogma of molecular biology, what we've talked about before. Other viruses might use something like reverse transcriptase to do this, such as the retroviruses like HIV, but SARS-CoV-2, indeed all coronaviruses, use a different protein. And essentially the messenger RNA is what codes for the protein sequence, right? And so the messenger RNA carries the code and essentially tells you which amino acids you string together to make the polypeptides that then fold into the viral proteins. And then the proteins will then self-assemble into the virus within the host cell. All right, so these mutations can impact which amino acids are used or not used, right? And this can impact protein structure and function, right? Because the shape is so important. And if you have a mutation where a different amino acid or a different sequence of amino acids is used, that could profoundly impact the shape of the protein and the function of the protein. However, it's also important to realize that it may not impact function in any important way, particularly if it's deep inside of the protein where there's not really a whole lot of interaction with docking with the cell and fusing with the host cell and so on and so forth, right? So not all mutations are gonna benefit the virus, and indeed most mutations are either inert or are just an evolutionary dead end, and then that particular virus that has a mutation cannot replicate, and that's just an evolutionary dead end. And indeed, that is often the case. But every so often, you will get a mutation that has some evolutionary benefit to the virus, particularly if there's some selection pressure being placed on the virus, like a vaccine that's causing certain kinds of antibodies to be produced. Then if you have a virus like SARS-CoV-2 that mutates, you get a mutation maybe once every 10,000 viral replications or something like that. Well, you literally have billions and billions and billions of viruses, and millions and millions and millions of viral infected cells that are replicating billions and billions of viruses. And so even if it's a 1 in 10,000 thing, you have the potential of getting lots of mutations occurring, even in just a single virus infected individual. All right. So with SARS-CoV-2, we are most concerned about the spike protein, and I've talked about spike protein in other videos. This is just a very important protein in terms of the viral docking and entry into the cell, and it's also very important as an antigen because it has so many epitopes. So the spike protein actually is made up of approximately 1,300 amino acids. So it is a chain of 1,300 amino acids that make up the spike protein. And in fact, I believe it's 1,273, 1,273 amino acids to be exact. All right. And what we can do is we can conceptualize the amino acid structure of spike by laying out the entire protein in a single line. This is not literally what the protein looks like, but this is just a way of conceptualizing it. So on one end, I will have the N terminus, right? So this is the NH2 that I talked about earlier, right? And then on the other end, I have the C terminus. And this, of course, is the COOH, the carboxylic acid of the very last amino acid. And then in between the very first amino acid or amino acid residue, amino acid number 1, and 1273, I have all the amino acids that make up different components of the spike protein. Of incredible importance are going to be a couple of domains that I'm going to talk about specifically. And there is this domain at amino acid 14 to about 306. All right, so over here in this area right here, amino acid residues 14 to 306 are what make up something known as the NTD, or the N-terminal domain, right? This is the domain that is very close to the N-terminus, or the NH2, the amine group, all right? And then another important area is going to include amino acids 330 to 528. And these are the amino acids that make up something known as the RBD. The RBD stands for the receptor binding domain. And the receptor binding domain is the part of the spike protein that actually binds with the cellular receptor. And just as a review, in the case of SARS-CoV-2, it actually binds with the ACE, the angiotensin converting enzyme 2 receptor. And this is highly concentrated in respiratory and respiratory epithelial cells, but we also see it in blood vessels and other organs and tissues throughout the body. So when we talk about the spike protein binding to the ACE2 receptor, it is actually this receptor binding domain component of the spike protein. So here's the thing. When I get mutations occurring in these areas, specifically in the receptor binding domain, certain mutations may be associated with increased affinity for the virus to bind to the receptor. So it's better able to bind to the receptor, and that might impact spread of the virus. It might make the virus more contagious. If that mutation involves an area of the spike protein in the receptor binding domain, and possibly even the N-terminal domain here, where you get epitopes where certain antibodies can bind and potentially neutralize the viral particle. If you have mutations in the N-terminal domain and the receptor binding domain that change that epitope, those antibodies may no longer be able to interact and bind to that area of the spike protein, and this may impact the ability for those immunoglobulins or antibodies to neutralize the virus. And of course, this can also impact the monoclonal antibodies, because they are essentially very specific antibodies that we produce therapeutically to bind to very specific epitopes And if those specific epitopes undergo certain mutations, those monoclonal antibodies might not be able to bind and neutralize the virus, right? So that's why this is so important. And the reason that I'm throwing out these numbers is it's important to understand that these numbers are in reference to the protein, or the amino acid number in this protein, in this polypeptide chain that makes up the spike protein. Okay, a really good way of conceptualizing this I found is in this article here. And I am using this photograph, and I want to be very clear that this is not something that I've done, and I'm using this for non-monetized educational content for purposes only. So think of this as a Creative Commons, and can be referenced. The specific diagram can be referenced in a very good article that came out in March 2020. And this article is called Cryo-EM Structure of the 2019 Novel Coronavirus Spike in the Pre-Fusion Conformation. So pre-fusion means prior to the spike protein, or prior to the virus fusing with the cell and essentially dumping its guts, its RNA, and its the proteins like RNA-dependent RNA polymerase into the cell. Right, so this is pre-fusion spike versus post-fusion spike, and a lot of these epitopes are very important when it comes to the pre-fusion, like prior to the virus docking and binding to the cell. So this is a really good article, and what they did is they used a procedure called cryo-EM, which is a really cool procedure. I'll just briefly talk about it. So the OG, the original gangster method of visualizing proteins is something called X-ray crystallography. So essentially what you do is you have to work very hard to get the protein to a very cold temperature, and you get it to form a crystalline structure, and then you crystallize it, and that essentially hardens it and makes it very tough. Because then what you do is you shoot X-rays into that, bounce X-rays off of it, and you can produce an image using X-rays. And a newer, kind of a groundbreaking procedure, it's relatively new, is called cryo-EM. That stands for cryogenic electron microscopy. So what you do is you essentially take the protein, get it to a very cold temperature, and instead of shooting X-rays at it, you shoot electrons at it. And so it is a modified electron microscopy technique, and it has really been somewhat of a game changer in molecular biology. So I figured I would just mention that because it is important. Okay, so this diagram that comes from this article is essentially the picture that I drew for you on the last slide. And so here I have the protein chain, the polypeptide chain. I have amino acid number 1 here. So this is the NH2 here. This is the N-terminus on the left-hand side, and then on the right-hand side here, 1273. That's the last amino acid that makes up the spike protein. This is the C-terminus. So this is the COOH, the carboxylic acid group of the 1273rd amino acid. And so this blue area here is the N-terminal domain that codes for the N-terminal domain. And then this green area here is the part of the polypeptide chain that codes, or that makes up the RBD, the receptor binding domain. All right, and then you have many other kinds of domains. You have S1 and S2, and essentially this is the two major subgroups of the spike protein. And in fact, if you look at it closer, the spike protein actually has 3 major groups to it, and we sometimes call these trimers, and it is a trimeric protein. We're not really going to go into that kind of detail. And then you do have some other areas or other fine-grained structures, CDHR1. You got the SS, or the signaling area. But again, what I want to do is I want to focus on the N-terminal domain and the receptor binding domain, the RBD, because these are so important. So when we look at these, when we look at the cryo-EM structure of the spike protein, this diagram here, so here's the viral membrane, and then deeper you actually have the capsid. I'll draw kind of the capsid of the virus here, and you have the RNA and the RNA-dependent RNA polymerase within the capsid, right? And then the spike protein coming out of the viral membrane. And so this green area here is the receptor binding domain right there. And then this blue area here is the N-terminal domain, and you can see that right here. And you can conceptualize the RBD as being kind of like the hand, right? So if you look at a hand, I'll draw this. So the actual fingers that grab on to the host cell. All right, so you can look at, maybe this is the cell membrane here, and you've got the ACE2 receptor there. The actual hand that grabs onto it can be conceptualized as RBD. And then the back of the hand, while it's not directly involved in the binding, obviously is a very important component, right? Because you need the back of your hand, because your finger's kind of attached to the back of your hand there, right? That the back of the hand is kind of the N-terminal domain, or NTD. So this other picture here on the right-hand side of the diagram is actually the spike protein has been flipped over. So imagine that I've rotated it like this, and I've rotated it 90 degrees. So you're now looking straight at it. So it is now rotated to where imagine that this part is now poking out of the picture, out of what you're seeing on your screen, pointing directly at you, and so now you're looking at it from on top. And so what we have here is you're looking directly at on top of the receptor binding domain, and then on the side here you're looking at the top of the N-terminal domain, right? So this is just another way of conceptualizing the spike protein. So you can see the pictures that we draw of the spike protein are often highly oversimplified, and indeed reality is often much more complicated and messy than the conceptualized pictures that we draw and that we make, just to help us develop some intuition. But again, hopefully this is helping you to better understand the structure of the spike protein and what's going on. Okay, so here we are, finally getting to the crux of this video. So when talking about mutations, let me stick with green here since I already have blue. So when we talk about mutations and deletions, right? So a mutation is essentially you mutate and you are replacing something for something else, amino acids, or deletion is where you knock an amino acid out. So when we talk about these, this is the nomenclature that you're often going to see when it comes to mutations. All right, so you're going to see something like this, and this is actually a major mutation that's found in the B1.1.7 lineage, also known as the UK VOC, variant of concern. So this is the variant that was originally isolated and sequenced in the United Kingdom. And one of the substantial mutations in that variant is the N501Y. So when you see this, the N is the original amino acid, and then 501 is the position where that amino acid or that amino acid residue is found. And so this would be at 501. So this is 501 out of 1273, right? And so if you go back, that tells you that you are talking about this is somewhere in that RBD, that receptor binding domain, versus the N-terminal domain. All right, so N501, and then Y is the code for the amino acid. So N is the code for the original amino acid. N in this case is asparagine, and Y is code for what that original amino acid was replaced with. In this case, it was tyrosine. So this essentially says this mutation is an asparagine at 501 was replaced with tyrosine. That is what that actually means. That is what that specific mutation is telling us, right? So this means that asparagine at position 501 was mutated to tyrosine. Now, deletions have a slightly different way of being conceptualized. And a common way of writing it is either deletion H69-V70. All right. So again, H is code for an amino acid at position 69. So this, instead of being in the receptor binding domain, like the mutation up here, right, this is in the N-terminal domain. So you have a deletion of an amino acid at position 69 of H, and I'll talk about what all these codes mean. And you also have a deletion of another amino acid, V, at position 70. Occasionally, this is written as delta H69-delta V70. Delta just means deletion. H is code for histidine, whereas V is a code for valine. These are, of course, just amino acids. And this is yet another major component of the B117 lineage. So not only do you have a mutation within B117, a mutation N501Y found in the receptor binding domain, but you also have a deletion found within the N-terminal domain of B117. All right, so again, this is just how this is just the basics for what it's meant when you see a specific mutation or deletion. So I'm going to include a table here, and again, this table can be referenced via this link up here. And again, I'm using this as a Creative Commons and a non-monetized video for its educational content only. So these are the one-letter symbols. A is alanine, R is arginine, asparagine is N, aspartic acid is D, cysteine C, glutamic acid E, glutamine Q, glycine G, histidine H, isoleucine I, leucine L, lysine K, phenylalanine F, and so on and so forth, tyrosine Y, valine V, and serine S, and so on and so forth. All right. The goal of this video is not to memorize all these, but just to know what these are talking about, right? So if you see, for example, let's say Q306H. I just completely pulled that out of my butt. What does that mean? That means that the glutamine at position 306 was mutated to a histidine. Okay, so let's put all this together and let's talk about some of the mutations that we see in some of these variants. Of course, the B117 variant has two major mutations and a deletion. So there is a mutation D614G in 501Y, and then the deletion H69 and deletion V70. And it is thought that this mutation here, the 501Y, may increase the spread or the contagiousness because that is impacting the receptor binding domain. That receptor, the viral, the RBD of the spike can bind more effectively or more efficiently to the ACE2 inhibitor. When we look at the variant that was originally identified in South Africa, this is the B1351 variant. It also has the D614G mutation, but it has a couple, and the N501Y, but it has a couple of substantial mutations, the E484K and the K417N mutations. And these impact, of course, the receptor binding domain, and these may actually have impact on the monoclonal antibodies such as BAM-BAM or bamlanivimab. In addition to that, the K417N can also might impact the effectiveness of another of the the monoclonal antibodies, the casirivimab, and then the imdevimab up here in the E484K mutation. So it does appear, I think there's fairly decent evidence that suggests that the monoclonals may be far less effective on this variant. If we look at the Cal20C variant, this is the variant that was originally sequenced and identified in California. There is a substantial mutation there, the L452R, where it may have decreased activity to BAM-BAM. And we may have decreased antibody activity in people who have prior infections, right? So they may be more prone to being reinfected with this specific variant. If we look at the B11248 variant that was originally, I believe, sequenced in Japan, but it was on a patient from Brazil, if I recall. So the Japan/Brazil variant of concern. It also has the N501Y mutation, and it also has the E484K mutation. So this may also have increased ability to spread, and it may have activity against or have resistance to some of the different monoclonal antibodies. Now, this concept of mutation is nothing new, and in fact, when we look at HIV, this is actually an important part of HIV and HIV therapy. There are many different antivirals out there, and typically we'll do a triple suppression therapy. You'll have 3 different antivirals or an antiviral cocktail that patients go on. And HIV is not an RNA virus, or excuse me, it is an RNA virus. It just uses reverse transcriptase. But there are many different variants of HIV, and these variants have different mutations that impact the effectiveness of these antiviral agents. And so knowing the mutations in the virus that the patient has is going to be important to selecting the proper antiviral cocktail for that patient. And it really does appear that something like that is emerging with SARS-CoV-2, where the different monoclonals that we use, and then perhaps even the immunizations that we use will be impacted by the different mutations and deletions found within these variants. And again, it does seem that the The receptor binding domain and the N-terminal domain are very important components of this because, again, this is where the lion's share of the binding occurs, and there are many, many different epitopes in these particular areas of the viral spike protein of SARS-CoV-2 virus. Okay, I think I'm going to cut it off here. This is probably going to be a very long video, but I hope you now understand or better understand what is going on with these mutations and deletions, and you better understand what all of the lingo, what all of the what it actually means, and what it means when you see a specific mutation, and what the terminology actually means. Okay, I'm going to cut it off here. I hope you all are doing well, and please stay safe and healthy.